Let's take a close look at just exactly how that whole coagulation pathway works. So here you see it all starts with prothrombin being formed via either the intrinsic or extrinsic pathways, all right? Triggered by tissue damage, involves many procoagulants and clotting factors. The goal of both pathways is to form prothrombin activator, all right? So, and you need to know every single detail of those pathways. No, I'm just kidding. You don't. I wouldn't do that to you. If you take more advanced classes in anatomy and physiology, you might. It's not uncommon to be required to know that. But all I want you to know just is that due to damage to the tissue and to the blood vessel wall, there are a couple of pathways that produce what's called prothrombin activator. That's all you know. You don't have to know the steps. But look at all the things involved. I mean, just point out all those clotting factors. Notice calcium is involved, platelet factor down there, PF3. So it takes a lot of steps. Try to imagine what would happen if you were missing one of those. Like what would happen if maybe you were born with a genetic defect and you didn't have clotting factor number 11, for example. We'll see that later. That does happen, and it has a name. You might even know it already. So what happens here, prothrombin activator, so let's follow through the diagram. Prothrombin activator converts prothrombin into thrombin. Prothrombin is a plasma protein. It's always in your blood, all right? It's one of those ones made by the liver. So it's out there, and normally it doesn't do anything. It just hangs out in your blood. But when prothrombin activator is formed due to injury to the tissue and the blood vessel, then prothrombin activator will start turning prothrombin into thrombin. Remember, thrombin, root word there, thrombus means a blood clot. So thrombin is what's going to make the clot. So how does it do that? Well, once again, fibrinogen is a plasma protein. It's in your blood right now. It's just in an inactive form. But once thrombin has been formed, so prothrombin activator, due to tissue injury, turned prothrombin into thrombin. Now thrombin will turn fibrinogen into fibrin. And fibrin is basically the magic step. Fibrin is what makes that big mesh, that big gooey net. That's what's going to catch all the blood cells and the platelets and actually form the clot. So prothrombin activator catalyzes the transformation of prothrombin to the active enzyme thrombin. Thrombin converts soluble fibrinogen into fibrin. See, fibrinogen is soluble. It's a plasma protein, but fibrin is not soluble. It actually precipitates out of the blood to form the net. Fibrin strands form the structural basis of the clot. Fibrin forms a gooey net for formed elements, including red blood cells, white blood cells, and platelets. Factor 13 cross-links with fibrin and stabilizes the clot. That's the final step. Once again, a clotting factor. I got that Rona. Imagine what would happen again if you didn't have factor 13. You couldn't stabilize the clot. All right, so there you go. There's the epic movie. There's the whole thing. Again, I just want you to know that basically there are a couple of pathways that in response to tissue damage and blood vessel wall damage, they form the prothrombin activator, and then the following steps after that. All right? So a hemopoietic stem cell will give rise to what? Erythrocytes, leukocytes, platelets, all of the above, or none of the above? Hopefully this is an easy one. All of them. The stem cell makes all of them. And what determines which ones it makes? Well, for erythrocytes and for platelets, it's a hormone. For leukocytes, it's colony-stimulating factors in response to the presence of an infection. And there you see a blood clot, white blood cells, red blood cells, platelets, all stuck together in that big giant fibrin net, okay? Platelet party! What's going on here? Platelet party! Why, I haven't had this much fun in years. Platelet party! I think we're stuck. Platelet party! No, seriously, we're stuck. Platelet party! Yeah, that's what happens. Remember, it's a positive feedback loop. Those von Willebrand factor and everything makes everything all stick together. That's the platelet plug. Now, given that you made a clot, do you want it to last the rest of your life? No, you don't. You only want it to stay there until you can repair the blood vessel wall and any other damaged tissue. So we have to have a process whereby we can get rid of the clot. Clot retraction, fibrinolysis, and tissue repair. So actin and myosin, do those sound familiar? Bio-201, muscle proteins, the contractile proteins. Those are inside platelets. They start contracting within about 30 to 60 minutes after the platelet plug forms. So as they contract, they pull on the fibrin strands, squeezing serum from the clot. Then, platelet-derived growth factor stimulates the division of smooth muscle cells and fibroblasts to start repairing the blood vessel wall. So in other words, once the clot has stabilized, you start secreting this growth factor that stimulates the growth of the repair of the blood vessel wall. Vascular endothelial growth factor, VEGF, also stimulates endothelial cells to multiply and restore the endothelial lining. So PDGF, VEGF growth factors, those are the growth factors that help you repair. Those are part of the whole process. You have to repair the injury, and then we'll be able to get rid of the clot. Fibrinolysis usually begins within a couple of days. Fibrin lysis. Lysis means break down. We're going to break down the fibrin. So plasminogen is another one of those proteins in the clot, is converted to plasmin by tissue plasminogen activator, TPA, factor 12, and thrombin. So TPA, by the way, is something that they'll give people after a stroke, all right? Things to try to help dissolve clots. So plasmin is a fibrin-digesting enzyme, and that's another example of homeostasis, all right? You had all the processes in order to form the clot in the first place, and then we have a series of processes to break down the clot and restore us back to normal again, okay? So notice that thrombin plays a double role here. Thrombin helps convert the fibrinogen into fibrin to make the clot, but then later on, thrombin will be turning plasminogen into plasmin, which will help break the clot down. So that little pathway, that little schematic diagram in the upper right, that's really good. It shows the whole deal there. Shows how, first you form the clot, then notice thrombin is in both pathways because later it stimulates the breakdown of the clot. Okay, so that's clots. Let's talk about transfusions, all right? Let's taking blood from one person and putting it into another person. Whole blood transfusions are used when blood loss is substantial, all right? Serious injury or in an operation or something. Packed red blood cells are used to restore the oxygen carrying capacity. So, for example, when my wife had cancer and she was undergoing chemotherapy, the chemotherapy kills a lot of red blood cells. The thing is, she wasn't short on blood. She still had her normal blood volume. She just didn't have enough red blood cells. So instead of giving her whole blood, they gave her what are called packed red cells, basically just erythrocytes, in order to help increase the oxygen carrying capacity of the blood. Now, transfusions have, people thought about that, of course, a long, long time ago. You see somebody losing blood, you think, gosh, maybe we get somebody else's blood and put it in. Well, that works sometimes, and sometimes it doesn't. There's a lot of trial and error here. Transfusion of incompatible blood can be fatal. And believe me, a lot of people died before they figured out exactly how to transfuse blood. I mean, in a way, it was worth it. If they were bleeding to death anyway, you might as well give it a shot. But in some cases, it wasn't actually, whatever originally caused the injury that killed them. It was the transfusion that killed them. Notice in the lower right there, if you're in the hospital and you're getting blood, you don't really like to see a mosquito on the line. Let's talk about human blood groups. You've heard of these before. Let's figure out exactly what they mean. So red blood cell membranes bear 30 types of glycoprotein antigens. So antigens are little surface markers. You have tons of them. All the cells in your body have tons of little antigens. Antigens can be from bad guys, or you can have what are called self-antigens. And self-antigens are what we have on red blood cells that determine our blood type. They're perceived as foreign, however, if transfused blood is mismatched. So if you give the wrong type of blood to somebody, if they don't have the right glycoproteins on them, then your immune system thinks that that blood is a foreign pathogen, a foreign invader, and it will attack it. And that's when the transfusion reaction happens. That's why it's so critical to make sure to give a correct transfusion. Otherwise, the immune system will spring into action to try to kill what it perceives as invaders. So promoters of agglutination are called agglutinogens. So antigens and agglutinogens turn out to be basically the same thing in this case. Agglutination means clotting, forming together, clumping together is what it means. And that's what causes the transfusion reaction. Your immune system will grab a hold of all these improperly transfused blood cells, and they will form a big clump, and then that forms a clot, and you end up dying because you have a clot that can potentially end up going to your lungs or to your brain, causing a pulmonary embolism, a stroke, and so on. Presence or absence of each antigen is used to classify blood cells into groups. So here we go. The antigens that are most commonly talked about are the ABO and the RH. There are actually tons of different kinds of antigens. There are lots of ways to classify blood. There's the MNS, the Duffy, the Kell, the Lewis, and so on. We don't talk about those very much because those turn out to be very weak agglutinogens. So even if you do a mismatched transfusion with those, it's usually not a big deal. However, if you mismatch the ABO antigens or you mismatch the RH antigens, you get a vigorous transfusion reaction, and that's potentially life-threatening. So look at those blood cells. See how there's the glycoprotein. Look at the type O. See the little hexagons there, a green one and a yellow one and a blue one. That's the glycoprotein. Type O just has a naked glycoprotein. Type A has the A antigen. It's an acetylgalactosamine. But in other words, that's what makes it type A. It has that particular little molecule sitting on the glycoprotein. Type B has a different molecule on the glycoprotein. And if you have type AB blood, you have both glycoproteins. So do you see? That's how we categorize blood. Type O, because there's nothing there. That type O is really zero. There's no antigen. Type A blood has the A antigen. Type B blood has the B antigen. Type AB blood has both A and B antigens. So this is what you have to be careful of when you transfuse blood. Now, types A, B, and O, that's the presence of the agglutinogens or the antigens. Really, either term is okay. Now, what's happening here is that your plasma normally contains antibodies. Antibodies are what grab a hold of antigens, and they handcuff them. I think I mentioned that before. They don't actually kill anything. They handcuff them together. Now, the deal is that if you have type A blood, this lower diagram is the diagram that I have pasted and copied and pasted, I guess, into my own head. Whenever I have to solve a blood transfusion question, I close my eyes, and I see this diagram inside my head, and this diagram will solve every blood transfusion question you need to solve about the ABO groups. So what happens is if you're born with type A blood, your plasma has anti-B antibodies. If you're born with type B blood, your plasma has type A antibodies. If you're born with type AB blood, you have no antibodies. If you're born with type O blood, you have both the A and the B antibodies. Notice essentially you have the opposite antibodies to what your antigens are. So if you have type A blood, you have the B antibody. Type B blood, you have the A. If you have type AB, then you don't have any, and if you have type O, you have neither antigen, so you have both antibodies. Notice that you couldn't possibly have an antibody for your own antigen. Otherwise, you would clot your own blood. Do you see what I'm saying? So type A blood could not possibly have an anti-A antibody, or you would clot your own blood, all right? So what's happening here then is we just need to, by the way, these things are not present at birth. That's why transfusions are a little bit safer in newborns. They form later. So earlier I think I said they were there when you were born. So it's really at around 2 months of age. But look at how to solve problems. It's so easy. So let's say I ask you a question. Can somebody with type O blood receive a transfusion from somebody with type B? Okay, let's think about that. If you have type B blood, you have the B antigen. If we put that into somebody with type O has both antibodies, right? So the antibodies in the type O blood are going to grab the type B antigen. So no, you can't do that. You cannot put type B into a type O person. You'll create a transfusion reaction. What about, could you put type B into a type AB person? Well, take a look. Type AB has no antibodies. So yeah, you can put type B into a type AB. In fact, who can you put into type AB? You can put any type of blood into type AB because type AB doesn't have any antibodies. That's why people with type AB blood are called the universal recipients. They can receive any type of blood. Now, how about this? Can you put type O into a person who's type A? Well, let's think about it. Type A person is going to have those anti-B antibodies. What about type O? Type O has no antigens. So yeah, you can put type O into a type A. Doesn't matter that the type A person has B antibodies. There's no antigens on the type O red blood cell. In fact, you could put type O blood into anybody, couldn't you? Because there are no antigens on the red blood cell. That's why type O is the universal donor. Now, you can memorize these things if you want to, but I think that's a ridiculous waste of brain space, and I think you'll probably forget and get it wrong anyway. It's much easier just to figure it out every time you need to know it. It's so easy to figure out, all right? So practice a few of those. Now then, that's the ABO group. There's also the RH blood group. There are at least 45 different RH agglutinogens, or RH factors. C, D, and E are the most common. That really doesn't make that big of a deal. But RH positive is the designation we use for people who have the D antigen. That's the problem one. Why is it called RH, by the way? It was originally discovered in rhesus monkeys. The RH is for rhesus, okay? So what you have to make sure of here is, take a look at that diagram there. So when you see, we have the ABO designation, and then we also add the RH designation. So O negative means you have type O blood, but you don't have the RH factor. O positive means you have type O blood, and you do have the RH. See, get the difference? Type B negative means you have B blood with no RH factor. B positive means you have B with the RH factor, okay? So what you have to make sure of here, once again, is if you're transfusing blood, you have to make sure that you don't put an antigen into somebody's blood who has antibodies. So who has the antibodies? Well, in this case, anti-RH antibodies are not spontaneously formed in RH negative individuals. Remember, an RH positive person couldn't possibly have antibodies, otherwise they would be attacking their own blood. It's the RH negative people. But as it turns out, they don't normally form antibodies unless they are exposed to RH positive blood. So if an RH negative person comes into contact with RH positive blood, then they'll form the antibodies. So what does that mean? Can you put any type of RH designation, negative or positive, into a person that has RH positive blood? The answer is yes, because if you have RH positive blood, since you normally have the RH antigen, you couldn't possibly have any antibodies. RH positive people will never have antibodies. So it's always safe to put either RH positive or negative into an RH positive person. What if you have an RH negative person? Well, if they've never come into contact with RH positive blood before, then it would be safe to transfuse them with RH positive blood. The question is, how do you know if they've ever been in contact with RH positive blood? Doesn't necessarily mean they've had a blood transfusion before, it just means they've come into contact with it. What if they play, rough sports and, like they end up, getting other people's blood on them? That might be enough. Rough sex. There are lots of ways that a person might have come into contact with somebody else's blood. So can you ever guarantee that somebody who is Rh negative has not been exposed to Rh positive blood? No, you can't. And so therefore, what is the rule? You will never, ever put Rh positive into an Rh negative. That's the easy to remember rule. That's how you keep this one straight. You never, ever put positive into a negative. You can put anything into a positive because they definitely don't have any antibodies. Negative, you don't know if they have antibodies or not, therefore you never tempt fate. You never put Rh positive into an Rh negative. Second exposure, of an Rh negative person to Rh positive blood will result in a transfusion reaction. All right, you can't ever put positive into a negative. And by the way, there's this whole conspiracy theory that people with Rh negative blood are not from planet Earth. They're actually the offspring of aliens. It's really cool. If you want to waste a couple of hours, just Google that and delve into it. There are people who actually believe this. All right. My dad died when we couldn't remember his blood type. As he died, he kept insisting for us to be positive, but it's hard without him. So sad. Blood is thicker than water. It can also clot and cause sudden death. Do you have, I used to have this aunt who had all these sayings, well, what's good for the goose is good for the gander, and all these other incomprehensible little things. But I loved it. If she would ever say, blood is thicker than water, I would look right at her and say, yeah, it can also clot and cause sudden death. That's a good way to shut up your crazy aunt. Blood is thicker than water, but maple syrup is thicker than blood, so in conclusion, waffles are more important than family. Keep that in mind. Waffles more important than family.